Hello, my name is Dedan Githae, a PhD student in the Jagiellonian University's Małopolska Center of Biotechnology in Krakow, Poland. I want to present to you a poster from one of our work titled To Reuse or Not to Reuse, Nanopore Sequencing as a Forensic Tool. The study of microbial communities, the diversity and the abundance has been used in the field of microbial forensics as a way of gaining an insight and intelligence of their ecological niche. With the advent of long-read sequencing platforms such as PacBio and the Oxford Nanopore sequencing technologies, they are quickly becoming popular due to their relatively low associated cost of sequencing. In addition, long reads help overcome challenges encountered from the short read, high throughput sequencing approach. For example, resolving incomplete assemblies due to complex genomic regions. In this project, we aimed to study the usability of the Oxford Nanopore flow cells for forensic applications and the implications of subsequent reuse to identify stable baseline microbiomes in an environment. Libraries from a soil sample were prepared and thereafter sequenced in 3 intervals, each 24 hours apart using the Oxford Nanopore Technologies MinION sequencer. One additional sequencing run was performed 1 week later. This calling of the raw signal was done using GAPI version 3, and then the sequenced reads were then taken for further bioinformatics analysis. We observed that the reads from different time points gradually reduce over time. The metadata associated with these sequencing runs obtained from the sequencing logs also showed decreasing number of active pores during subsequent reuse. The reads obtained, we used Kraken version 2 for taxonomic identification of these reads, sequencing reads, and then re-estimated the taxonomic abundance at the species level and at the genus level using Kraken. Our findings, we found that the number of identified taxa differ between the different time points, with the bacterial abundance and diversity reducing after subsequent sequencing runs. These taxa that were lost also happen to be the lowly abundant taxa present in the environmental sample. However, the majority of the microbial communities that were present and identified in all the time points had little variation between them in terms of abundance over the different sequencing time points, as shown in the diagram. These abundant bacterial communities give a stable descriptive baseline of the environmental microbial profile of the niche of interest, which makes us say that nanopore sequencing technology offers a viable approach for environmental metagenomic study and application in forensics. And from this, the underlying microbial signatures can be used as a stable way of distinguishing between different environments. Thank you for your attention, and I invite your questions and comments.